Okay. So welcome. Again, my name is Emily Ferguson, and we're going to do botany today. The uh, thing that you should know to begin with is that botany is usually a 3 month long semester course in colleges. So we are going to be dabbling in botany today. And it's going to feel a little bit like a um, really bad car ride. So, um, welcome. I'm your driver, and hopefully, along the way, we will take some breaks to help you not feel like you are sitting in a Zoom room for too long. And if you have questions along the way, please unmute yourself and ask me, or you can put it in the chat, and I will try to answer it as we go along. This is being recorded, but we want to make sure that you guys get what you're looking for and need out of this program. So we're going to start with the fact that it's been pretty cold recently. So there aren't a lot of new pictures to show everybody because a lot of it's covered with snow and ice. So when you're thinking about botany, I want to start with acclimating our minds to what do you need to be able to do botany? Things you don't need are any degrees because this really is all about a passion, an interest, a curiosity. Those are things that are so much more important to be able to pick up and play with and enjoy botany. An interest in the natural world, the ability to use your senses, observing things, and having an interest in sleuthing and looking for answers when you see something you don't understand. So we should start off. What is botany? It's from the Greek botane, meaning plant, and it really is the study of patterns in plants. And can you guys give me any patterns that are in plants? I'd like this to be a little interactive if at all possible. And you can type it in the box or you can say it out.
So the patterns in plants are the seasons and just the growth.
Yes, so that's perfect. The seasonality that goes with plants and things that happen repeatedly every year. So this time of year, a lot of them don't have any flowers. There's a really good reason for that. And then when we get to spring, we have flowers, then we have the formation of fruit, then we have growth through the summer, we have fall colors, we have all these different things that are happening. And in botany today, someone's put in leaf patterns where seeds arise. That's good stuff. So here are some of the patterns we're going to play with today. We're going to look at structure of plants. First, we're going to have to do with cell structure. Then we'll go through the origin of plants, classification, naming plant parts, how energy is developed and moved, identification of plants, and reproduction. And the idea is to give you a little bit of sort of a touchstone on some of the important pieces. I know you guys have your handbook and you had a chapter on botany. I got to look at that too, so that they gave me some sort of guidelines of what we should be making sure you get a touchstone on today while we're here. So plant cell structure. What kind of organisms are plants? Plants are multicellular, and as you can see from this picture, you can see lots of cells on this piece of this leaf. And we have to talk about cell theory. There are two different types of cells. There are prokaryotic cells and there's eukaryotic cells. Prokaryotic cells are usually unicellular and lack a nucleus and membrane, any membrane-bound organelles. So things like bacteria that are small and simple are prokaryotic cells. Eukaryotic cells contain a nucleus and other membrane-bound organelles. And the idea in cell theory is that all living things are made up of one cell or more cells, And cells are the basic building blocks of life. And all cells come from some pre-existing cell. Not sure about that last one, but it's kind of part of the cell theory that came up. This was a theory formulated by German scientists in the mid 19th century. So um, the take home from this slide is that everything has cells, and we're going to be focusing on the eukaryotic side of things, not the prokaryotic side, because we want to go further than bacteria today. There we go. So we have to compare plant and animal cells and see what the differences are between them. Plant cells have to perform two functions that aren't required by animal cells. Our plants have to produce their own food, which they do through photosynthesis, and they have to be able to support their own weight. They don't have a skeleton like an animal does. So, because of those two things, they have a couple of different features that are part of their cells. And here is the plant cell, and the organelles that cells have that animal cells don't have to support those functions are chloroplasts, which are these green things in here, the cell wall that goes all the way around the cell, and then usually this huge, big, it looks like a pool here today, a kidney bean shaped pool, but it's the large central vacuole. And we'll get into why we have all those. So the structure of the cell is made of the cell wall is made of cellulose, and that gives support and structure. And that cell wall can also be, because of that cellulose, things can go through it in one direction or the other. The large central vacuole that's in the cell is going to make sure and give the cell turgor pressure. So when that's full, and it helps support and hold up the plant. That large vacuole also will help maintain the pH of the cell so that we don't get too acidic and we don't get too basic. And that vacuole is also used for storage. So water is stored there, nutrients, pigments, as well as waste. And for anybody that's done any plant classes before, the anthocyanin, that's the red in the fall that we see, and the purple in our blueberries, that's manufactured um, from pigments that are in this large central vacuole. And if our large central vacuole doesn't have enough water in it, we call it a flaccid cell. And if our cell is flaccid, that means that our plant will not stand up. It will be wilted. And I have a peace lily that tends to really like to show me when I have not given it enough water. But when there's enough water in a plant and that water enters by osmosis, that vacuole is going to swell and it pushes against the cell wall. And then we have turgid pressure, and that helps keep the support and structure there. The next part in our plant cell is that our plants are autotrophs, so they produce their own food. And they do that through the chloroplasts, and that's where photosynthesis takes place. And that's where the pigment chlorophyll is, which absorbs all the wavelengths except for green, which is why we see green in plants. So now that we've done cell theory, we can get on to different things like the origin of plants. So here's the origin of plants. And they started off as a group of green algae. And that's where all plants come from. This is a simple cladogram or diagram that shows how things broke off over time. So we have the green algae here at the beginning, and then the next part of the tree is where bryophytes came off, where we have mosses, and that contains hornworts and liverworts as well. And then the next branch is where we have seedless vascular plants, and that's going to be our ferns, horsetails, club mosses, spike mosses, and quillworts are all in that group. And then we're going to have the gymnosperms as the next piece that comes off, and that's going to include our actually our conifers. Do you guys want to give examples of those? Because you might know those better than me, or at least can be able to participate here. We're looking for conifers. Pines, good. Um, Fir trees, cypress, junipers. Um, what are some unusual gymnosperms? Do you guys know any unusual ones? We have some gymnosperms that lose their leaves. Um, so there's the larch, and there's also we have the ginkgo that's in this one. Bold cypress, good. Ooh, nice job. And then finally, we have the angiosperms, and these are going to be the flowering plants. And that's going to be they angiosperms with they have covered seeds, and they produce flowers with an ovary, which develops into fruit. So when we start off this, we have mosses that are not doing very much in the way for reproduction, and don't have any vascular tissue, so they can't get really tall. And then we go into the ferns that start having vascular tissue, but they still need water for reproduction. And then we go to our gymnosperms that have got height and ability to get nice and tall because they've got some organization and the ability to create wood. And they actually have those naked seeds. And then we have our angiosperms that have the covered seeds. And we're going to see that a little bit more as we go along. So this is another rendering of what we just talked about, but it has more with each piece. So we have our non-vascular here. Then we have things that have vascular plants, and those are going to be those also produce spores here. And then when we go down to our superdivision, we have the naked seeds up here, and then we have the angiosperms down here. And to really drive home that point, we have our mosses here that aren't able to get very tall. And then we have our ferns that now have vascular systems, but they still have spores. So these are non-vascular spores, and we have vascular spores. We want to, that transition there is from being able to be really small and tiny to starting to get large and big. Because I think we all know that in the Jurassic period, we hear of tree ferns and really large um, lycopodiums and things like that. And then we have our gymnosperms. They have the naked seed. Here in the mountains, we have Table Mountain pine. We have these baby megastrobili or seed cones that are starting on this new growth section of this pine. And I think it takes 2 to 5 they can produce 2 to 5 cones each year, and those cones remain closed for 2 to 5 years after they've matured, and only about 40% of them open without fire, because this is a fire-dependent species. And then we have the pollen cones that are male, so micro, strobili. Micro is always male, mega is always female in the plant um, system of words that they use. And the word strobili here is the reproductive structure that is holding a number of modified leaves or ovule bearing scales, and they're grouped into something called a cone. And these pollen pieces are these pollen cones, they come up every year, and they are the things that turn our cars green with pollen. And I hear a lot of people talk about having allergies to pine trees. I've read different accounts of whether or not they can be, you can have allergies to them. Some things say you can, and some things say you can't. So I'll just go with there's a potential to be allergic to anything. So just be careful. And then we have the angiosperms, which have the vessel seeds or the covered seeds. And we have different examples of them here. We have the tall bellflower, we have the trillium, and then we have the portfolio bellwort. And these are all going to be wildflowers that we find in this area. Does anybody have any questions thus far? All right, I guess that quietness means no. So we're going to go on to biological classification. And to do that, we have to first talk about Linnaeus. He's the father of modern technology, taxonomy. Can't be technology because that would be wrong. So he was a Swedish naturalist, physician, and botanist, and he was granted nobility in 1761. And he developed the hierarchical system of classification for nature. And it started off as kingdom, division, class, order, family, genus, species. For plants, we usually use the word phylum instead of division. And this is taxonomic hierarchy. And the entire classification originally was created by him, which I think is pretty cool. So he named plants that were sent to him from all over the world, including North America, in a document called Species Plantarum, 1753, and he classified plants into logical categories to show their relationship to one another. And he based a lot of these on structural similarities, particularly within their reproductive parts. So when we look at flowers on plants, different flowering structures are often distinguished between different families. We'll see as we go. And you can see here at one point he was using class and order. Class was for the number and length of stamens, so those are the male parts of the flower. And he used order as the number for the number of pistils, and that's the female part of the flower. So that's how he sort of divided things out as he was doing taxonomy. Um, so when he talks about structural similarities, um, we have to be careful because there's different ways that things can structurally be similar. We have things that are homologs, which is where they share an embryonic origin that's similar. So the fact that we have a hand, a bat has a wing, and a whale has a flipper, those are all homologous. They're all the same bone structure underneath, just in different formation or pattern. So we all came from a common ancestor. Analogous are things like the bat of a wing, the wing of a butterfly, and a bird wing. While they're all different groups, like bird, insect, mammal, um, they all have the same function. So when Linnaeus was doing all of his categorization of everything, he may not have understood whether it was a homologous or an analogous structure that he was considering similar. So there's a chance that he may have gotten things wrong along the way, but um, that's how it goes. And I think it's really neat. I was reading up for teaching today, and actually, I'll save that. I don't want to tell you now. I'll keep that for later. But it's really important for us to think about the structural similarities and where they arrive from and how we get to where we are now, or whether we developed something because we needed it, even if we're further down the chain. So here's the original classification. The domain is the top, and it starts with eukaryota, which is great because that's for eukaryotic cells, which we talked about at the very beginning is the origin of all of our plants, and all of us too, because we have animal cells versus plant cells. And we're going to be looking at plant cells today. The kingdom is Plantae, and then the subkingdom is Tracheobionata, and then we have the Spermatophyta, and that's going to be the superdivision. Those are where the first seed plants are. Then we have Coniferata for the pine side of things for the phylum, and Magnifoliata for angiosperms. And then The next piece, class, is in red, and I'll show you that in a second. But as you get down here, we have class, then subclass, order, family, genus, species. And we tend to sort of live in this genus species area, but there are some people who like the family organization to help them get to the genus species. So I tend to be one of those people, and I will look at the family taxon um, and start there for identifying a plant, and then go further down into genus or species. But I tend to start at the family level because I look at the big picture and then zero in rather than be zoomed in and then try and back my way out. Uh, and as I said on the bottom here, any of these group levels are referred to as a taxon and plural is taxa. So all of these together are taxa, and each one is a taxon. And that class piece, that class taxon that I just mentioned, that's where the angiosperms are divided into two different groups, the monocots and the dicots. There are nowhere near as many monocots as there are of dicots. And our monocots don't always get to tree level, and that is because of their vascular bundle arrangement. So I'm going to quickly go through these, and we're going to talk about each part of these today. So they'll become more familiar as we go. Seeds are different. Monocot means one seed leaf, and there's only one leaf in or one seed leaf in the seed. And dicot means two seed leaf, which is why there's dicotyledon. After that, the names don't really help, but things for dicots for the flowers are usually in multiples of five for the floral parts. For monocots, they're usually in multiples of three. I think this is four or five, I should say, for dicots. Sorry. The venation of leaves for dicots is usually net-like. It's usually parallel for monocot venation on leaves. We're going to skip the pollen piece, and we're going to go to vascular bundles. In monocots, they're sort of distributed at random around the inside of the stem of a plant, and because of this random distribution, they're not organized in any way in order for this plant to become strong enough to become a tree. And if you think about monocots, mostly people think of grasses. The two that get to tree-like are going to be bamboo, which is just a really large grass. And the other one that is in the monocots that gets to tree-like is going to be a palm tree. The dicots have this circular vascular bundle array that goes around the stem, and because of that, they're able to get to woody structure, and we'll get into that later on. But because of that, they are all the trees that we see around us that are not gymnosperms. And I've put the classification for each of these for a trillium on one side and for campanula over here. And sorry, I looks like I got a little squished. So over time, so Linnaeus, did all that work in the 17th century, which was a while ago. um, there was a bit of a drift in classification, and different botanists started using different systems. And there's an Egger system in Europe. There's the Bentham and Hooker system that was used in Britain, and there was a Russian system, and then in the United States there was a Cronquist system for classification. So Linnaeus started it, and then these guys started sort of doing their own thing where they were. And all the original classifications were on flower parts and some biochemistry if they had it, not always. But then came along phylogenetics. And phylogenetics is the study of how organisms have evolved over time and the relationships within them and within the organisms and within the group. So things changed, and they started changing in 1993. when they started using morphological traits and photosynthesis genetics to be able to understand or to sort of map out plants. So they're now using, um, say, they're using a flower type, but they're also using the photosynthesis gene in that flower to figure out where it lands within a classification system by the photosynthesis of it, um, and the photosynthetic gene and how it's made up. So it's becoming less about what the morphological traits are and more about what the DNA shows. And to be fair, at the beginning, it was using just the photosynthetic gene, which is going to be um, in the chloroplast. So it's a little bit tricky. They're using a mixture of two different things. So they started this, and the first angiosperm phylogeny group classification came out in 1998. Angiosperm phylogeny continues to be taken care of today, and I'll get to that in a second. So there's an APG one, which was 1998. We've had a couple different renditions, and APG 4 came out in 2016. And Peter Stevens is one of the authors for the APG, and he maintains the website, which is the Angiosperm Phylogeny website, which I've hyperlinked to this here in case anybody wants to go and look at it. It's hosted by the Missouri Botanical Gardens, and he's been regularly updating it since 2001. Um, and it has a lot of the useful information of what they're doing and how they're doing it. So the principles are sort of the same. So they want to maintain Linnaeus's classic system of orders and families, but they want to make sure that the groups are monophyletic, and that's where the game changer comes in, because a monophyletic group has to consist of all the descendants of a common ancestor, and that can sound pretty crazy. So we're gonna take a look at that a little closer. So monophyletic versus polyphyletic versus paraphyletic. And these diagrams are simple cladograms, which is a line diagram that branches suggesting phylogenetic relationships among organisms. And the organisms are arranged in such a way as that they share a common trait or characteristic, or that they don't share with other clades. So I will not have all the same information as B in this one over here, but B has a relationship with everybody below it, and everybody below it came or originates from this B here. Does that make sense? I hope I see some nodding heads. Um, Here, this would, so these guys might be twins for this person. So this would be your grandmother. Your grandmother had your dad and your sister, and then your dad married somebody and had two kids, and you had two kids, and you're all related. Does that make sense? Okay. In this one here, this is polyphyletic because E and G are not related. So this would be where we took a trait like the wing of an insect and thought it was similar to the wing of a bird. Does that make sense?
I have a question.
Sure.
I don't understand why in the first figure, A,
Hmm.
E and G are in the monophyletic group, but they're
Right.
not monophyletic.
Oh, so here they are because the whole purple is outlining the monophyletic group. Whereas in two, this is only, they're thinking that just these two individuals are in this group and they're polyphyletic because they're not related. The idea is to get back to the common ancestor. So for us to circle just E and G, that would be incorrect because they're not related. But if we
Oh,
went back far enough, we get to B, and B is related to F, C, D, E, G, and H. But here,
okay,
if I
so it's
just
not
circled,
It's not about the descendants, it's about how the group is defined.
Perfect. Yes.
Okay,
So
got
the
it.
group has to be
Thanks.
defined from this point out to the end. Um, because if I start at A and I put a circle around this whole thing, the whole thing is monophyletic. But this is monophyletic in this instance. This is mixing families. So, um, or so this would be like saying that your niece and your son are related. Um, and that's the only two that are related. Like you'd have to go back further to the grandmother to get everybody who's related to E and G. Does that make sense?
Yes, it makes sense to me. Thanks.
Okay, and then here in this last one, paraphyletic, is where we capture one group, but we forget everybody else over here. And these guys should all be included. So we've sort of narrowed our focus too much, and we didn't get the breadth of everybody that A has a relationship to. Um, so because this isn't all of the descendants of A. This is just a portion of the descendants of A. We need all of these guys to make it the full thing. But the point of this graph is to say that because of all of this new technology and testing with genetics, a lot of the plants that we thought were related are not as related as we originally thought. So things have been if you meet an old timer botanist, they will complain that, they've messed everything up. And there might be some truth to that, but I'm not going to get into that piece today. But the idea is that because of all of this new technology, we're getting a better sense of who's related to who and we're getting that more correct. And that's the important piece from all this genetic information. And it gets really complex really fast. But I just wanted to sort of show you uh, we're in the dicots here, and then we travel along, and we've got the core dicots, and then we go up here, and when we get over to an order, and then we have a family over here, and then within these families would be genus and species. So here we're seeing in the here's the rose uh, order, and then we have all the families here, and that obviously includes the roses. It includes elms. It includes the figs. It includes uh, the weed family, cannabis family. So all of those, they're not closely related, but they're all coming from this Rosalie start. Does that make sense? I hope so. The piece that you should keep in mind is that this is still in flux. So they're still doing experiments. They're still learning about this. They're still doing better research. They've moved on from using just the um, chloroplast DNA. They've come back and they're starting to use mitochondrial and nuclear DNA. So all the testing that they're doing for this phylogenetic stuff is getting stronger and stronger support. But for right now, when you learn genus and species names, um, take them with a grain of salt because there's a chance they might slide and change. But as you can see, they've been doing this for over 20 years. They're getting pretty good at it. So we shouldn't have to worry about a lot more earth-shattering changes. Let's see if we can get to that. So the current classification, when you look at it, so they wanted to keep order, family, genus, and species, but instead of having those different, it used to be kingdom, class, phylum, like kingdom, phylum. Yeah, I can't do it. I can say the acronym, but I can't get all the things that are the same. But instead of having phylum, division, and all those other things, suborder, subclass, they're missing here, and they just use the word clade. So they're maintaining the order and families listed with the principles of phylogenetics, but they've removed the layers of the original Linnaean classification system, which makes it a little bit harder to talk about everything, but as long as I get families, I'm pretty happy. So the next thing we want to do is talk about plant names, and they can have common names, and those can be misleading. Does anybody recognize any of these pictures and want to talk about them being misleading? And I can even give you all their names if you want. There's an oak tree. It's a live oak from Florida in there. Yep. Oh, moss. Yes, that's Spanish moss. But is it really a moss? And the answer to that is no. It's actually a bromeliad, which is a monocot, and it's the problem. This is what I'm trying to illustrate: is the common names can be a problem because first you think it might be from Spain, which it's not, and then second you think it's a moss instead of an actual angiosperm. So that changes where it is in the origins of plants. So that can be a bit of a trick. Mountain ash in the top right or top left, that one's sneaky too, because this is not a mountain species, or at least I don't see it growing in the mountains, and it's not an ash tree either. It's actually in the rose family, and it's more closely related to your rose bush in your yard than to an ash tree. And then hop hornbeam and hornbeam, they have some very common names, which helps everybody get them confused. Um, Hornbeam has several other common names like musclewood, blue beech, or water beech. And then they both have the common name of ironwood. And there's a lot of places that have trees that are called ironwood. So it can get really confusing really quickly. I have friends in Florida and California, even internationally, and they'll send me a picture of a plant and say, "Is this ironwood?" And I'm like, "Oh, I don't know, but I can. Maybe we should switch over and use the Latin names or the botanical names because that I might be able to help you with better." But it's important to know your audience and where you are so that when you do help with an identification of something, you get it right. So the importance of common names versus botanical names. Sometimes it's easier using common names, especially when you are at the nursery or talking to friends. And I totally get that. I'm not going to go up to my mom and talk to her about her um, philodendron spa because she's going to be like, "What are you talking about?" when she wants me to just call it elephant ear. Um, but we should try to use our botanical names, scientific names, or the binomial nomenclature. And here I've listed this out. The genus is usually a noun, and that sometimes reflects the organism's relationship with other organisms. And then the specific epithet is usually an adjective or a possessive noun, and it describes either geographic range, habitat, part of a plant, or it honors a person. So, and I should talk about when you see a Latin name, and I'm gonna just, ooh, I did a very bad job of that. I should probably undo that and try again. Okay. So usually when you see this, the first letter of the genus is capital, and then the second word for the specific epithet is usually lowercase. And if it's been written by a person, it's usually either in print and then it's underlined, or if you have the opportunity, because you've typed it, usually italicize it. And then the L at the end of this, that is the person who originally named it. And in this instance, that was Linnaeus. So when you see the L by any of these for a botanical name, you know that Linnaeus is the first person that identified it and named it. And this is Hamamelis virginiana. Hamamelis means together with fruit, and that refers to the idea that simultaneously you have flowers and maturing fruit from the previous season on the same tree. It's a Greek word, and Virginiana is going to refer to the habitat of, or the geographic range of where this was previously found and discovered. Does anybody have any questions about these?
I have a question. Could you give an example where the specific epithet is part of the plant? What does that mean?
Oh my gosh, you are a plant. This is perfect. I'm going to go to the next slide. So, when I have to get rid of that thing that I drew on there. Okay, so here we have three different plants. We have Spring Beauty, Claytonia virginica, Nyssa sylvatica, which is black gum, and flowering dogwood, which is Cornus florida. And I think um, sylvatica means growing in the forest, so that's a habitat. But Florida in Cornus florida means abundant with flowers. So that is a description of the plant. And there's some things, Cephalanthus, that's for buttonbush. Cephalanthus means big head. And that's because it has that big circular flower ball on it for big head. And then in this instance, Spring Beauty, Claytonia, is honors a colonial Virginian botanist, John Clayton, and the Virginica is where it's found. Is anything with Virginica native? Most likely, if it has Virginica, it is native. Um, and the only caveat to that is when Linnaeus was naming these things, that was back in 1753, right? Virginia was a very large state at that time. So you might find things that are more Pennsylvanian or West Virginia, Kentucky that are no longer Virginica, even if they have that as their specific epithet. Does that make sense? And then I should mention up here, I put this note on this slide so I would remember the International Code of Nomenclature convenes every 6 years, and they provide guidelines for the naming convention of plants. So one of the things that's really important is that the person who discovered it first is the person whose name gets to stay with it. Um, and the so if so Claytonia virginica was named by Linnaeus. If um if John Clayton had come to the United States to do some botanizing and he found it and called it Veronia alba, I hope that's not really a species. Um, John Clayton wouldn't be given if those two names were both for the same plant at the International Code of Nomenclature, they'd say, John, I'm really sorry, we can't call it that. Linnaeus called it something first, and he takes priority and precedence because he was the first person to discover it and name it. So the other name will become a pseudonym, something that's sometimes used, but we try to not use it anymore. And I hope that makes sense. I just got asked a question in the chat. With the new phylogenetics, are the genus names changing? And the answer is yes. When I was in grad school, the aster genus, which is what the Asteraceae is named after, um, pretty much got demolished. And because of the International Code of Nomenclature, they found an older name that genus had been previously named Symphyotrichum. So all of the asters are now Symphyotrichums instead of aster. And the nice thing is that almost every text or book will still have those old names because um, people that get involved in botany stay around and are enjoying and part of that community for a very long time. And it's really hard for some of them to let go of those older names. So we have to try to be all encompassing when we use plant names. And I think you'll find if you look at the Flora of Virginia, when you look at the app or even in the book, you will find those pseudonym names or those older names listed after the current name that is agreed upon. And there's a lot of people that complain about the name changes, but it's important because it demonstrates our advancement in knowledge for botany. So it is kind of cool to see that. Any other questions on naming?
If they provide the older names once the genus or other um, taxon is changed, do they also provide common names or they just don't have any use for those?
Nope, they will put common names in there too.
Okay.
Um, because and they try very hard to capture the most common of common names. I don't know if that the most widely used common names, but some colloquial names may not be captured. So when you look at the flora, you'll see some of those things in there. So it is. Oh, and then here I put I actually annotated with what each one does, but I forgot to put that in earlier.
Oh, I have
So,
old field guides. Sorry. So
oh,
I
that's
just looked
okay.
at it. I was looking to see if Astor got changed, but I looked at the date. It's in 1977, so it's not going
Uh,
to show anything.
but the floor of Virginia would show that. And so I decided, because you guys are all sitting at home, that we needed to create some labs where you got to get up and go somewhere and do something. Otherwise, you're going to be at your computer forever and hate me later. So, does anybody have any house plants, artificial or real, around them that they would like to pick up or look at and tell us if they're an angiosperm or a gymnosperm? And then we could figure out if they're a monocot or a dicot, and if you know their botanical name. Oh, someone's got a flower. Hold on, I'm going to pause this because I want to see. Ooh, you've got is that a Dieffenbachia, Debbie?
I noticed a spider, whatever.
Oh, I love this. People are bringing their plants to the computer. This is fantastic.
Yes.
This has worked much better than I thought it would. Um,
So I don't know what the the spider plant, spider lily, whatever you call them. I don't even know the
well,
common
so
name.
so I think while that has wider leaves, I think that's going to be a monocot because I think it has um, that sort of peace lily flower to it. Is that right?
Yes.
Yeah. Okay. Good. And Amanda, what do you have there?
A begonia.
A begonia. Ooh, that's a good one.
Very
And
pretty.
they're pretty fleshy, aren't they?
Yeah, and they have like the little tuberous rhizome spreading roots underneath.
Ooh, good. We're gonna talk about those later.
And I have a couple different varieties over here. So
And do you think a begonia is a monocot or a dicot?
I mean, it
And that
does
one might
get
be tricky because it
flowers.
has those funny flowers, doesn't it?
It does get little tiny pink flowers on it. So
Okay. It's gonna be a dicot. Just
that's what I was thinking, but
excellent. Good. This is going well. Tracy, can you tell us about your plant? Oh, you have to probably put in the chat. Those look like
yeah,
palmate leaves. Is
Monstera
that
deliciosa.
ooh, Monstera? That's monocot too. I love that you got the whole name for it. And Monstera usually has holes, naturally occurring holes in its leaves, right?
The fenestration between them.
Yeah. And those holes are so that water goes through the leaf to the roots instead of off the leaf to another plant. It's very sneaky that way. Tracy, I think you might have is that uh a geranium? Okay. Could you type it in for us? And that peace lily that Patty put in, that's definitely monocot. And I love the fun fact from Diana. Yep. I love the fact that Spanish moss is related to pineapples. So cool. Pelargonium. Oh, I don't know that. Ooh, that specific epithet suggests that it has an odor. Does it have a smelly flower or a fragrant flower? Yeah, good stuff. Um, how about Jenny? You got a plant there.
Yeah, this was a gift I got just a couple days ago. And it
Oh,
has the little, plant thing in it. And on one side, it's called Superba Fittonia. And on the other side, they say Fittonia superba.
oh, now that's interesting.
Yeah,
Um,
I haven't looked it up, obviously, so I don't know.
I always thought those were like pepperonia,
That's what I thought too.
but
Pep-
that could be one of those ones where they've changed the Latin for it.
So,
Um, Very cool. And that from those reticulate veins on those leaves, I would definitely say we have a dicot there instead of a monocot. Phyllis, what have you got with you?
I'm actually not sure, but I think it's a
Oh,
type of
great.
coleus.
Oh,
It was also a gift.
so so for coleus, do you have a square stem?
Um, Square stem.
Yeah, you should be able to put your fingers around the stem and it should sort of click between your fingers instead of roll.
Oh yeah, yeah, it is. Oh, that's
Okay,
so
so you
cool. I never noticed that before.
Good. Yeah, all of your things in the Coleus family, Lamnaceae are gonna have square stems. Um,
Yeah.
oh, so uh, a green onion? That's awesome. Who's got the green onion growing? Oh, and you've got another uh something in the um philodendron family there
This
with you,
is the
Amanda.
neon philodendron.
Yeah, very cool. An arrowhead plant. That's Trish, right? Uh, and do you have the space on those when you get a flower? No. Okay. Well, I didn't really know how well this would go. This has gone much better than I thought. Ooh, Diana, do you have something else that you're showing us? Oh, you're on mute. You're gonna have to unmute yourself.
It's a Christmas cactus.
Oh, very nice. Pulling out the cactus. And that's a great one because that one uses the light in order to know when to flower, right? So if you want to make sure it flowers on time for Christmas, you want to make sure that it's getting
Really?
those
Okay.
longer nights and shorter days. So if you keep it under a lit
Really?
light in your living room too many hours of the day, you're not going to get as many flowers as you expect.
That's good to know. Thank you.
Yeah, and poinsettias are the same way. They need that. they need to be able to measure how much light versus dark they're getting in order to know when they're supposed to be flowering.
That's why I love plants. So
They
interesting.
they are very sneaky and very conniving. They know all sorts of ways to get around us, which is
Fascinating.
why I like
Thank
them
you.
too. Um, Anybody else have any other plants? I even listed artificial because I wasn't sure like how many people like where everybody was be sitting. But we'll get back. Um, ooh, Mary's got one. Hers looks like a jade plant. It is. So we're in another cactus. Oh, and she said something, but I can't hear her. It looks like she's got two in there. Oh, is there an aloe in that plant pot too? Good. I'm glad I can recognize plants through Zoom. This is a new technique I didn't know I had. Um, I'm gonna go back to sharing and get back to our thing. So I had no idea how this was gonna go, so I put my own house plants in here. Um, so I put in a philodendron here, and then I put in a Nepenthes because I really like to have carnivorous plants around my sink. So in the spring, summer, fall, when there's fruit flies, they have a place to land and go. Um, and then I have an orchid here, and I have a Hoya as well. And the philodendron is definitely a monocot. So mine are all angiosperms. Um, the Nepenthes, it's kind of cool. I had to look that up. I wasn't sure if it was going to be a monocot or a dicot, but carnivory has is an analogous trait. So it is found in both monocots and dicot species, and they think that it's evolved at least 6 different times independently, which that is just wild. And somebody who gave the fun fact about the bromeliad and the pineapple, Spanish moss and the pineapple, one of the newest carnivorous plants that they've found is also a bromeliad. In that well where they hold water in the middle, they've found that they release chemicals and toxins that can kill anything that lands in that water, and then the plant can digest it. Okay, then my orchid is going to be a monocot, and then my Hoya is also a dicot. Okay, thank you for participating in my lab. And does anybody need like a real couple minutes away from their computer to go and for a comfort stop? Yeah, let's take, can we take till 2:10 for comfort stop? Okay, I'm going to pause our recording so that everybody can go and do that, and we'll be back. Hey, welcome back. And now we're going to do the anatomy of plants, plant parts. And I'm going to throw in the physiology for growth and development as we go because it's really hard to pull those things out. So they're going to sort of be folded into this piece. And I like to start off with the seed. And to be completely honest, it wasn't until I took a course like a year and a half ago that I even learned that a seed has its own nomenclature and names for the outside of the seed, which has the seed coat, which is really called the testa, and then the scar where the seed was attached to the ovary is called the helium. And then there's a micropyle, which is an opening where water can get into the seed. And if we had been in person, I had a lab for you to do on this. But since you're not, we'd had our alternate lab instead. And then the inside of the seed is going to have this little funny shape here, which is going to include the epicotyl, which is at the top, right below where the first two leaves are. and then we have the hypocotyl in the middle, and then the radicle will be at the bottom. And these are just parts of the stem that are all sort of mushed together here. And then these are the embryo leaves, and then the cotyledon is all this space in here. And that's the stored food for the embryonic plant. And it's important for us to recognize when you, if you guys have peanuts at home and you open up a peanut, you can see that the embryonic um, plant in there, and you can see two leaves, and you can see the epicotyl, hypocotyl, and radicle all smushed together in there. And then everything else that you're eating is the cotyledon, which was the stored energy for the embryo plant. And that's kind of cool to do. You can't really do that in classrooms anymore because there's too many kids that are allergic to peanuts. But if you're at a ball game and you're bored, you can entertain the person next to you by these new cool facts. Or you can maintain your friendship and some space from people by not sharing that. Germination is something that's going to happen to the seed, but while the seed coat is on, it protects the dormant embryo until it's ready for germination. And germination is going to only occur when certain conditions are met. There needs to be ample water, there must be optimal temperature, and there needs to be a location with aerated soil. And depending on the seed species, some will need a cold period, a stratification, and others might even need scarification. Um, and scarification often is done by us by either nicking the seed or throwing it in a blender. What is that? What is that emulating that we don't see? What is happening in nature that does that when we have to do it in a blender or nick it ourselves? Does anybody want to guess?
Birds.
Right. So something going through the esophagus of an animal or the stomach of an animal. So birds, mammals, anything that is eating seed, and it has to go through their intestines where there's a pit of acid and digestive juices that are going to scare across that seed and nick it in order for it to be able to germinate. So all of those answers are correct. And stratification for cold period is obviously emulating winter. So our dicot germination, I just wanted to throw in a slide that shows this. First out is going to be the radicle, then we're going to have the hypocotyl, and then the hypocotyl is going to allow out the epicotyl, and we're going to have the cotyledons. Actually, I got it wrong. The epicotyl is going to be above the cotyledons, and then there's the pumila, or the first full leaves. Notice that we have the cotyledons above ground, and then we have the first true leaves. And that can be different depending on the species. Here's a monocot seed. Didn't want to leave them behind. They only have one seed leaf, which is going to be in here. And then they have the cotyledon around it, which is different. And it's only one cotyledon. And then here is their germination piece. And it's really interesting when we talked earlier about plants being autotrophic because they produce their own food. This is the only time in their life cycle that they're heterotrophic, when they need that endosperm, those cotyledon, in order to actually be able to make it. That reliance on the endosperm and the cotyledon, where the energy is stored until it breaks the surface of the soil, they're heterotrophic because they're eating from another. They're not self-reliant, which is what happens once they get above ground and chlorophyll starts doing the work to produce sugars for the plant. And this is obviously the germination of a monocot. And then there's epigelial and there's hypogelial. And that is whether or not the cotyledon leaves become above ground or not. So in epigelial, they come above because they're epi, they're above the ground. In hypogelial, they stay within the soil and they don't come above ground. So those cotyledons stay down below. So maize, wheat, and peas do this. Whereas on this side, for the epigelial, we have sunflowers and castor and bean that do the other. So when you plant the famous bean seed that everybody makes you do in a classroom as a kid, um, those cotyledons come up first, and then you get the first pair of true leaves, which is why when somebody tries to send you a picture of a plant and it's only got two leaves to identify, usually it's really hard to identify that plant because we actually may not have true leaves yet. So then we want to talk about the plant parts. We have vegetative parts, and some people refer to these as organs. The leaves that are going to be photosynthetic, the stems that are going to be using or that are going to be doing storage, growth, and transport. And then we have the roots that are going to be absorbing and anchoring and storage as well. And then the reproductive parts of the plant are going to be the flowers. So we're going to focus on the vegetative parts for a little while, and then we'll get over to the flowers. And we'll get closer to some of these pictures that are on this side. Yeah, and I won't get anything away, go too far with that. Maybe I will say, for me, when I'm trying to remember xylem and phloem and what's going up and where it is in the plant, and we'll get to xylem and phloem in a little bit, I think of X marking the spot for xylem in the middle of the root. And then it's going to be coming up on the inside for the vascular cambium. And then when it gets to a leaf, it's on the top side of a leaf. So that's how I remember where it is so that I don't get confused when someone hands me a plant or a slide and asks me to identify where I am and what it is. I usually have to keep that in mind to do it. One thing that we should probably try and smush in here are plant tissues. Roots, stems, and leaves have three different types of tissues. They have ground tissue, they have vascular tissue, they have dermal tissue, and they have one more that they're missing. And the cells and the tissue types are going to be parenchyma, collenchyma, and sclerenchyma for ground tissue. For vascular tissue, we have tracheids and vessel elements or sieve tubes and companion cells. And then dermal tissues, we have epidermal cells, stomata, and trichomes. We're going to go into more detail on that stuff, but I wanted to lay the groundwork and have a sort of a pictorial to show this to you. The missing tissue that's not listed here is the meristematic tissue, and that's going to be the apical, and so that's at the very top of the plant and the very bottom of the plant. Lateral meristems, which are going to be the vascular cambium and the cork cambium, and then the intercalary cambium. And these are all places where cells are being created that are new, and they're being done via mitosis. So they're creating exact replicas. They're not creating something for reproduction. So here are the ground tissues that I just mentioned, the parenchyma, the collenchyma, and the sclerenchyma. The parenchyma is photosynthetic, and it's also used for metabolic reactions and for food and water storage, and that's a potato there. For anybody that's trying to guess what the slide picture is. Sclerenchyma is for support, where growth occurs, and you can see they've got their potato peeler out and they're taking off the sclerenchyma on the celery stalk. That's the more chewy part that's harder to, that's stringy, that people don't often like to eat. And sclerenchyma provides structural support and protection. So it's usually found around seeds, in nuts, and they're often dead. So those are the ground tissues that are going to be in plants. And they're in roots, leaves, and stems. And we'll touch on where they are found in those places, but I wanted to at least give you the background so that you know where things are coming from. So here are roots. I like to start from the bottom up. Roots are the first part of the plant to emerge from a seed. Their function is to anchor and hold plants in place. They absorb, conduct, and water, and store water and mineral nutrients from the soil. They store excess food and nutrients, and they usually have connections with mycorrhizal fungi. And those can be ectomycorrhizal or endomycorrhizal. And ecto is on the outside and endo is on the inside. And funnily enough, that means on the inside or the outside of root cells. It doesn't mean inside or outside of plant root, which it took me a little while to get my mind around that. And we have an apical meristem at the bottom of this, and that's where the protoderm for the epidermis is, which is going to be the outside, the ground meristem, which is going to be the inside cortex, and then the vascular cambium is a procambium, which is going to be this piece here. And root hairs are going to be important in increasing the absorptive surface area on roots so that they can get more in there. I should mention mycorrhizae fungi have recently been discussed a lot in different venues about people talking about plants communicating with one another. So I took a class on this, and there was talk about mothering trees and things like that. The information so far scientifically has shown us that mycorrhizae will take nutrients from one plant and pass them along. So far, we have not seen another plant uptake those things and use them. So we're halfway through that story, but I think once we have more scientific proof, we'll get further on that piece. And I think that was someone out of Oregon who did that research, that grounding research. Suzanne, somebody, I'm sure somebody has read her book. But I don't like to go further than the science has shown me, so that's where I am. So here's a general picture of a root, and then it's important to talk about the differences between roots. We have monocot roots that are fibrous roots, and those are to occupy a large volume of soil, shallow soil. So they're really good to use for erosion control, and they grow really close to the surface. Whereas dicots tend to have a taproot, which is one that goes really deep looking for the water table. And I think that's all I have right now on roots. We're going to slide into vascular tissue. So this is going to be inside stem. I should have probably had my stem slide first, but the vascular system does start in the roots and goes all the way up through leaves. So xylem conducts water, and it moves water and dissolves substances from the roots to different parts of the tree or the plant. And xylem cells are usually dead, and they don't require any energy to move things around. The phloem, on the other hand, is conducting sugars and nutrients and organic pieces from anywhere that photosynthesis occurs in the plant to where it's needed. It looks like I have an extra, I got the wrong O in photosynthesis. I'll fix that. Transport in the phloem cells is active and it requires energy. And we'll circle back to where that energy comes from shortly. But xylem is just moving water from the bottom up through, and phloem is taking it from wherever it's manufactured to wherever it's needed within the plant. So that could be going up or down in the phloem. And then I wanted to show you the vascular tissues that we have. So xylem cells I just mentioned are dead. The walls have lots of lignin in them for strength, and they have tracheids with vasculoprints and gymnosperms, and then vessel elements um, that are going to be in angiosperms. So depending if you're a gymnosperm versus an angiosperm depends on which type of these cells you have. But they have perforated plates at the ends in order for things to go up pretty quickly. And then on the phloem, these are live, and they have companion cells. Often these sieve tube cells don't have a nucleus and some of the other things that a regular cell would have, which is why they have this companion cell so that they can continue to function and live in order to maintain the movement of these things with that energy that they need. So here is my stem cell. It should have been, or stem slide, it should have been a little bit higher. So stems are used to move things and for support. So supports the leaves to receive the maximum amount of sunlight, supports flowers, contain the vascular tissue to conduct water and nutrients, and they also can be used for storage. And what we do here is we start at the very top. This is going to be the apical meristem up here, where new cells are dividing and being created. And then we have the protoderm and the ground meristem and the procambium. And then we start getting things actually in a cambium. And then as it gets bigger and older, we have more specialization going inside there. Herbaceous plants have little to no secondary growth, and secondary growth allows for the increase of girth. And most of the things that we talk about for stems, they talk about them having a negative gravitropism because they grow towards the light, which makes them phototropic. I have some questions about that, but we'll get to that later on. And then on the outside of the stem, which is what we see and probably is the best part for us, we have nodes that are right here. And this is a position on the stem where leaves or branches or flowers originate from. And there's usually a bud next to them. And then the space in between the buds is the internode. So those are pretty important, especially when we start talking about ID for plants. They're going to be asking you about what you see at the nodes, and you could also be asked about the buds. And then there's some adaptations that stems do in order to have asexual reproduction. So you can have a plant with stolons, and that's a stem that's on the top of the soil that's sort of grows out, lands, puts down roots, and has another plant form. You can have rhizomes that are underground horizontal stems. So it'll grow linearly under the ground, and then it'll pop up with a new plant a little ways away from the original plant. And then you can have suckers, and those are things that are growing straight from roots underneath in order to create new plants. And these are different adaptations, as I said, for making sure the longevity of the plant keeps going. And I think that they talk about the largest organism on Earth is a clonal aspen, and it has a name, and I don't remember it, I'm sorry. But that's all through suckers and asexual reproduction. And then the last thing we should talk about here in this section is different plant storage organs. And these are usually either going to be stems or roots that have these ability to store nutrients. So we have lots of different forms. We have the carrot with the taproot. We have the onion bulb, which has storage in here. Then we have the corm, We have the rhizome. We have the roots of a sweet potato, or it looks like a dahlia. And then we have the stem of a potato. And here is the bulb close up. So these are short and squat. They're usually underground. It's an underground fleshy stem, and it has scales where it stores stuff for the winter. And it's interesting because these are actual leaves on the outside, and inside is where the future flower would be. So think of your daffodils and your crocuses and things like that. So we're going to does anybody have any questions? Okay, so that brings us next to our next little lab that we have. I was curious if anybody has any of these in their kitchen and to see if we could take a look at them. So we're looking for a white potato, a carrot, an onion, sweet potato, or ginger. And if you don't have any of them, don't worry. We can share what our findings are from what we got. I'm going to stop sharing for a minute, see what people come back with. Oh, someone's got, is that a carrot? All right. It's not an orange carrot, but it's still a carrot. So you've got a taproot right there then. Um, and you should be able to find, um, so you have the top of it and the bottom of it. Oh my goodness, I've lost there it is. There we go. Do you have any root hairs growing out of that? Yeah, maybe. Okay, good. Because that's gonna, oh yes, good, very nice, excellent. Um, Anybody else have any of these storage items in their house? Oops, someone else has got a carrot. Very nice. Debbie brought back a bulb of garlic. That's lovely. And she's got a potato. Is that a white potato? It looks like it is. Oh, someone's got ginger. Oh my gosh. Everybody's got some great stuff. Okay. Um, so these gingers, are they a rhizome? Are they a stem or are they a root?
Isn't ginger a root?
Ginger is a rhizome.
Ah, rhizome.
Yeah, so you should see nodes and internodes. You should even see adventitious roots on one side of it, and you should see um, there's even a leaf scale on a ginger. Yep, she's got a node that's growing. Nice, Mary. I like that. Um, it's kind of cool. And I think someone had a potato too. Was that you, Debbie? So that is a stem or a root. And this one's sneaky, so be careful. Oh, I can't hear you.
They said a potato is a stem.
Yes, it is.
I
So
mean,
you should,
I found it hard to see that, but they said it's a stem.
it is a stem.
With
So
the
you
buds.
should, it has buds on it, and it has eyes, and it should also have, it should even have lenticels on it. If you look with a hand lens, you should see little dots all over it. And those are for gas exchange. So, uh, and the part that's crazy is this a potato, a white, russet, whatever you're holding, red, they're all stems. But if you grab a sweet potato, that is a root storage organ. So, oh yeah, there's one. Mary's got a sweet potato, and you can see there's a little bit of a difference. You can see there's got like they've got the little knob on the bottom and part on the top. So they're a little bit different from uh, our our regular white potato. And it's really funny. So I'll tell you a quick story. My husband, we had flower pots that had sweet potato vines in them because they can be very decorative and pretty in the summer. And at the end of the season, I was like, are you gonna get those sweet potatoes?" And he was like, "What? There's no sweet potatoes in here." I was like, okay, I'm pretty sure there will be. And he was horrified and amazed to see that he had sweet potatoes. He wouldn't eat them because they'd come out of the soil, but he's a work in progress. So, and he can hear me, so I'm in big trouble. Um, okay, I appreciate you guys. Brian, did you have any of those kitchen items? Oh, he has a carrot. Good, nice. He's got a taproot there. He's good to go. Okay. Excellent. Well, this was supposed to be a short one. Diana, it looks like you had a potato there in your hand. Oh, you have an onion. Nice. Okay, and is that a stem or is it a root?
Is it a root?
It's actually supposed to be a fleshy stem.
Really?
Yeah, weird. Very weird.
Wow.
And that's the
Um,
same with garlic.
yep. Uh, garlic is garlic is a bulb, so it's going to be in that fleshy stem category. Which underground stem with fleshy leaf scales. So, um, I have to say, every time I have to teach something on botany or a class on anything, I always learn something. So, thank you for my days of prep to make sure I really didn't get anything super wrong. So, we're going to go back to the presentation, unless anybody has any questions.
I have a question. So the onion, then if the onion is a bulb or a stem, then are these the papers that come off of it, are they leaves?
So they are a part of they call that the tunic, and underneath is the leaves.
Okay.
So I would say a very bad analogy is that you know how we have some barks that shed. Or the scales on the outside of a bud that sheds so the leaf comes out. I would call the tunic those things rather than a full leaf, if that helps us get that right. Okay, going back to
Thanks.
sharing. And... All right, excellent. Thank you for playing along. We'll get back into, we got through roots and stems, so we have to definitely go a little bit further. And the first thing we're going to talk about is that meristematic tissue. So we have apical meristems, which can be found at the tips of plants and at the tip tops and the very bottoms. We have lateral meristems, which I mentioned earlier, are the vascular cambium and the cork cambium. We'll look at those momentarily. And then we have this intercalary meristem. And from before I started training or getting ready for this presentation, I thought those were only in grasses. But they are also using them with dicots because they're talking about the intercalary meristem helping leaves go out as well as allowing the expansion between the nodes to happen in the growth season on a regular plant. So when you first have a bud that comes out and it has a bunch of little leaves all squished together, as that expands and those leaves get bigger, that's where that meristem is coming into play. So there's
So
a
is
lot of
this
people
also
that sort of,
referring to auxiliary buds and adventitious buds? Is this the same thing?
yes. So those axillary buds, those popping out and growing would be starting from an intercalary, and then if they become the very top of the plant, then they'd be the apical meristem. So if you went in and pruned down to that, if you pruned down to here, then this would become the new apical meristem. Does that make sense? Okay. So very interesting, because usually we talk about that as just being able to describe why lawns keep growing up even though you mow them down. It also applies here in dicots as well. So here's a cross section of a trunk of a tree. So in the middle is where we're going to see our xylem. And we have, actually, this is probably labeled as cord. No, it's not. So we have primary xylem, and then we have secondary xylem, and then we're going to have the vascular cambium in between, and that's going to be the cambium that's creating new cells through mitosis that go either in as a xylem to the center or out as secondary phloem. And then we're going to have a cork cambium after that secondary phloem. And that's going to be the other lateral meristem. And that's going to be putting cells to the inside towards that secondary phloem. And it's also going to be sending cells out to make that cork cambium and that bark layer. It's just fascinating that trees are able to do this. So the next thing I wanted to mention when you see this is as that wood on the inside of a tree continues to expand, there's pressure that's put on that bark cambium. So we have the vascular cambium in the center that's doing the phloem and xylem, and then we have the bark cambium that's putting on girth outwardly for the bark. I decided I wanted to go a little bit more in depth with that. And that bark cambium and what's produced from that, those are considered part of the dermal tissue. The cork and bark layer, we talked about tissues way back. But I have an American beech here and then a red maple. And the American beech, the periderm expands to meet the growth of the wood below it. So if my cork cambium or if my vascular cambium puts on an inch for wood for the year, that bark cambium is going to put on 3 inches so that it doesn't cause cracking or fracturing in that bark. And that's why our American beech always has a very smooth bark. A red maple is a really good example of where the periderm or that bark cambium can't keep up with that vascular cambium. So when they're younger, the red maples have very smooth bark, but as they get older, there's a point with which that bark cambium doesn't keep up with the wood cambium. So it starts causing cracks and fissures and fractures in that bark, and those are longitudinal. Yes, Amanda.
So we were at a client's house the other day, and there was a tree there that at first we couldn't really recognize. But as we looked further up in the tree, it had the bark that looked like a beech tree that reminded us of it. But the main base of it was starting to exfoliate. Is that why? It ended
That's
up being
exact.
like ended up being a sweet beech, a very large sweet beech.
Oh,
um,
okay. Um,
um,
and I'm not familiar with the sweet beech, but that's that's usually what happens. When you're doing tree ID and you're using bark, if you don't see the bark you expect on a big tree, you're supposed to look up into the lower branches to see that bark
okay.
trait or characteristic on those lower branches. Like a black cherry is a really good example of that. Down low, or when it's young, it has that smooth bark with the lenticel lines on it. And as it gets older, those lenticel lines start breaking, or that lower bark starts breaking up, and it starts looking like burnt potato chips or burnt cornflakes. And you can still see those lenticel lines on them, but they're not as obvious. But if you look up the
Okay.
tree, you'll find that smooth bark higher in the top of the tree rather than down low. So
Okay.
um, um, Yeah, so that's why I wanted to sort of show this picture because the American beech is set up so that those two cambiums work in harmony or together so that you always have a smooth trunk on that tree, whereas other trees that bark cambium not keeping up with the cork cambium is what creates those bark patterns that we use to identify trees. So I think it's pretty cool. And I'm not supposed to be doing bark or tree ID today, so I'm going to keep going. We're at the leaf, finally. So we've done roots, we've done stems, and now we're at the leaf. And here is a basic leaf piece. We have the tip at the top or the apex. We have a midrib in the center. We have veins. We have a margin, which is the edge of the leaf. We have the petiole, which is where it attaches to the twig. And we have the lamina or the blade, which is everything that is on the top of the leaf or is the leaf surface area. And this is a simple leaf because it's undivided, and the leaf blade which is not separated into leaflets, but we could have this blade cut or cleft or deeply lobed, and that would still be a simple leaf. So here it is in cross section. We have the cuticle on top, which is part of the dermal tissue. We have guard cells and stomata on the bottom. That's also part of the dermal tissue. And the epidermis is going to be part of the dermal tissue as well. But then everything in between here is going to be ground tissue and then vascular tissue. So we have this palisade layer here, and then we have the spongy mesophyll down here. And these two sections together are called mesophyll. And most of the chloroplasts are going to be in this palisade layer up here, because this is the top of our leaf and this is the bottom. Usually stomata are mostly on the bottom of a leaf instead of the top of a leaf. And then we have our vein coming through. And remember from my X marks the spot on the bottom, as we come up, we're going to have xylem on top and phloem on bottom, which is what we've got here on the diagram. So we've got that right. And that's what is in the vascular bundle or the vein in this. So this is the most recognized portion of a plant. And they're considered the shoot system of these plants. And their function is to contain chloroplasts and the mesophyll. So it's in the palisade and the spongy layers for photosynthesis and the continuation of transpiration through their stomata and then the storage of food. And stomata are pretty cool. Botany is the only I will loosely call it a language, where we actually name things that don't exist. Like a stoma is the air space between the guard cells. So I'm not sure why we do that, but it's done often. So I should point that out and be transparent about it. So in the chloroplasts, we're going to have photosynthesis happening. Oops, sorry, I thought I had more on this. So here we have light that comes in and it has a reaction. Light is the energy that enters the system. And then we have the water that's coming in, usually from that has traveled all the way from the root. And then we have oxygen being created and glucose being at the outputs. And these are going to happen in the chloroplasts in that mesophyll layer of the plants. And we've got the CO2, which is also coming in through the stoma, and the water and with the sunlight creating the glucose and oxygen. And glucose is obviously a sugar. So while it's great and it's created, it's not something that the plant can actually use. So we'll have to figure out how to fix that. And the energy that's created, this ATP is adenosine triphosphate, sorry. It's an organic compound that provides energy and it drives most processes in the plant. When you talk about that Christmas cactus that someone had and the aloe and the jade, they have created different photosynthetic systems known as the the CAM cycle, where they actually collect their carbon dioxide at night so that their stoma aren't open and allowing for water to evaporate away. And they do part of the process during the night in order to be more efficient with their water. So there's CAM cells, and there's also a C4 system where the chlorophyll clusters around veins for higher efficiency when they're creating stuff. That's a little bit more advanced than we wanted, but I wanted to at least touch upon the fact that while this is the basic system, we do have a little bit more advanced systems for different environmental pressures, which I think is something that your manual talks a little bit about. And in the chat, someone asked if you can ever see stoma with the naked eye. I would lean towards no, but if you have a hand lens, I think you definitely can see them. Yeah, I know that for a citrus plant, if you hold that leaf up to the sunlight, you can see pinpricks of light through it. But I've never looked up to see whether that is because of stomata that are open. So I'm gonna say that I don't know about that one specifically, but I do think you can see them with a hand lens. So we have that glucose, and that's great. And obviously you guys and I love sugar, or different forms of sugar. But that's not really helpful for the plant. So they have to use a process to break that sugar down into energy. And this is something that is going to go on in plants. So they're taking that photosynthesis cycle that we just did, and they're running it backwards. And this happens in the mitochondrial. So every cell in the plant can do this. They can do respiration. And again, we talked about earlier that phloem needs energy in order to move sugars and other compounds around the plant. And they're going to be using respiration in the mitochondria to help facilitate that. So we're taking that glucose and we're going to break it down, and we're creating 36 ATP in order to help with processes throughout the plant. This is a very a generic picture of this cycle. It can be very more complex if you really want to get into it. And I should show that there's two different sections. So if you don't have access to oxygen, you end up doing fermentation and lactic acid. So when we get cramped, tired muscles from running too much or too fast, and that lactic acid buildup is because we're doing fermentation because we haven't had enough oxygen in our system. Whereas over here, and when we have the oxygen present, the Krebs cycle is what's happened. There's an electron transport chain, and then we create these 36 ATP that help us do all these different things. So there's different pieces. I thought the side-by-side comparison would help. So photosynthesis produces food, and then respiration uses that food to create energy. Photosynthesis stores energy, respiration releases energy. This occurs in the chloroplasts only. This occurs in all cells. This releases oxygen, this uses water, this produces water. This uses carbon dioxide. This produces carbon dioxide. This occurs in sunlight, and this can occur in darkness or light. Just so that you have those things side by side. And because we're talking about photosynthesis and respiration, the next thing we're going to move on to is transpiration and translocation. So transpiration is that water movement through the plant. And has anybody ever made those that Christmas garland where you take a piece of construction paper and you staple it through the next construction paper and you make this linked chain that goes around your tree? That's what I think of as when I think of transpiration. I think of that chain from the roots all the way through the stem of the tree, out the branch, and up to a leaf. And the sun is on the end. And with the sun causing evaporation or transpiration, it's plucking out a water molecule from the top, and that moves that whole chain along and makes space for another water molecule to come in at the bottom in the root system. That's a very basic look at it, but it is the process of moving water through the plant. It's a passive process. There's no energy expended to do it, and it's transported through that xylem. Translocation is different. That's going to be the active movement, and it's going to require that energy, and that's the movement of those sugars from photosynthesis to other parts of the plant that need those things. And if they're not needed, then they're going to go into storage. But these things, so that's going to use the phloem, and the transpiration is going to use the xylem. So I've tried throughout the presentation to keep phloem and xylem on the same sides throughout. I may have gotten it wrong somewhere along the way, but that was done on purpose to try and keep active or inactive, passive transport versus active different and separated. And I wanted to go a little bit further into transpiration. And I'm going to start at the bottom because water is going up from the root in through and out through the leaves. So water moves into the roots via osmosis. And if you'll remember, osmosis is when there's a diffusion of water. So if you have lots of molecules on one side and very few on the other, there's this high concentration wants to move over to the low concentration to get to balance. So when you have that water molecule that's been pulled away and you've got that hole in the root, water is going to push in through osmosis to fill in that gap and to get things back to a homeostasis or a balance. And then it's going to move through the plant, tree, stem, through capillary action, and that's going to be with cohesion and adhesion. Water molecules generally have a little bit of a negative charge, so they're attracting each other because they want to be next to each other to get to positive. And that's what's pulling them through up through the plant. And then, as I said earlier, that sun's going to be on the top end of that plant, and it's going to be helping evaporate that water out through those stomata. And hopefully that's enough of that to make that make sense.
So can I ask a question?
Absolutely.
So the transpiration process uses the xylem, and the xylem is very rigid. Is that right? And then the phloem, or whatever you call it, is more like we think of vascular, like flexible and like a blood vessel.
So think of it as a vein, but things being able to go on both sides. So because that flexibility is still there for the xylem, not as much so because the xylem cells are almost all exclusively dead, whereas the phloem cells are alive, but they both have lignin to help strengthen them and keep them stronger. I would think of them more as a roadway where things can go in one direction on one side and the other direction on the other side. And in case of emergency, like Florida, you can use both lanes to exit everybody from the state.
So does the phloem, the movement there, also help with the xylem flow?
So sometimes, because but remember it's going to be actively moving sugars to where they're needed in the plant. So sometimes that could be up further in a leaf or down further in the plant. And there's also times when things are moved from roots back up to the tree. So think of this time of year when plants are starting to break dormancy. um, the xylem is going to have excess coming in through the phloem because that sugar and stuff is going to help break bud and put out new leaves. So you're going to have both phloem and xylem going up this time of year because you want all those sugars back up in the leaves or back up to help produce leaves and create foliage and flowers and fruit. But towards the end of the season, while the xylem is still going up, phloem is going in the opposite direction to take things out of leaves because we don't want to store anything in a leaf that's going to fall. How's that? Does that help?
Yeah.
Okay. And that's why, even though we should have Florida swapped around, well, no, I think it's okay because the phloem can go either way. And instead of people coming into the state, we would swap them and have them all exiting to go up to make sure we have sugars in the rest of the tree. um, but I hate to make them the two of them together are considered the vascular bundle, which is why I don't want to break them apart. So they're together throughout the plant and they're together out in the veins, which is a little different from our circulatory system, right? We have veins that are taking things out and we have other arteries taking things back. I'm not a person biologist, so I could have gotten that wrong. So don't listen to me on that one. But I think that some is oxygenated and some is not, and they're not in the same. Okay, we're going to go on to more leaves, unless there's other questions. Okay, so we talked about the regular leaf of an angiosperm. I should say something about conifer leaves. They can have needles. They can have blunt and flat needles. They can have scale and awl like leaves. And I find scale and awl like leaves can sometimes be together on our junipers, our eastern red cedars. And then we have some that are just awls, and we have like the cypress, and then we have some that can be yeah. So that we have different types depending on the different tree. And you could take a whole class and get a whole bunch of new names for some of these, which I did, and it was a little overwhelming. But these are fine terminology for just getting us through conifer leaf types. And then we have tropic movement. So we've talked about roots, we've talked about stems, we've talked about a little bit about leaves. Tropic movements are the growth movement in a direction of a stimulus, and that's positive tropism. So when you grow up and towards the sun, they talk about a growth movement away from a stimulus being a negative tropism. And I'd love to get some examples of that because I hear that, gravitropism or is or I hear something about negative tropism for roots growing away from light, but I don't. It's kind of a little bit wishy-washy to me. Some common types of tropisms are gravitropism, phototropism, big tropism, that's for touch, so that's going to be a vine twirling around something, and hydrotropism, which is a response to water. So some things don't like water at all, and other things really love water. And I put in this picture of skunk cabbage because it has contractile roots. that are pulling it further down into the ground. So I would say that is a positive gravitropism if I was looking at that stuff. Um, so that's one type of movement. And then there's another type of movement plants do that is nastic movement. And this is a movement of a plant or a part of a plant in response to a change in temperature. Um, so rhododendron is often called nature's thermometer because at 40 degrees Fahrenheit, the leaves are open and horizontal. At 35 degrees Fahrenheit, the leaves start drooping. At 32, they are cupped and they are on the verge of curling. And at 25 degrees Fahrenheit, the leaves are curled very tightly. So why is this happening? And does anybody have any answers to this for me? And while you're thinking about that, these moves were figured out and seen by J.D. Hooker when he was in the Himalayas back in 1849. Conserve water is one possibility. Any others? So there's three hypotheses to deal with the stress of winter. One is snow load, lack of water, and the other one is often intense light. That wasn't supposed to happen that way, but that's okay. And the part that's really weird is that two of those hypotheses, lack of water and snow load, have been refuted already. So the often intense sunlight is the one that's last to be sort of standing. And experiments have shown that leaves that were forced to stay horizontal during the winter experienced permanent sun damage and photosynthesized considerably less than leaves that were allowed to droop once favorable temperatures returned. Um, and then they figured out that drooping and curling are two different types of environmental stress. And the curling leaves don't thaw out as fast as leaves that are left horizontal and flat. So by slowing down how fast they thaw, they avoid damage effects from freeze thaw cycles. So there's two different things happening. One is to avoid too much sunlight and damage the photosynthetic machinery, for lack of a better word. And then the curling is to prevent thawing too quickly. So if the leaf was flat and open, it would heat up and warm up, and those cells would maybe burst from that happening too fast, right? So all of us have tried freezing a leafy green, and then we pull it out of our freezer and it's, complete mush. And that's because it thawed too quickly. Therefore, like everything burst open and it just became a mush. Um, it's really interesting. There's so many things that we still don't know about plants, but I think it's pretty interesting to see that they're still figuring things out in a really good way. Okay, and then I thought I should compare movements. So tropic movements are unidirectional responses to stimulus. They're growth dependent and they're usually permanent, and they're found in all plants. Whereas nastic are non-directional responses to stimuli. They're growth independent of movement. They're only found in a few specialized plants, and it's usually immediate action. Do you guys have any examples that you might be able to give me?
Would the Calaitha plants, the prayer plants be an example?
Yes, I think they're a nastic example because they close and open that's not permanent. What else closes and opens that you guys know of? Sensitive plant, yes, I would say that's also a nastic. The mimosa, Venus flytrap, that's a nastic movement. Um, and what's really interesting, it's usually a species that we know, like prayer plant, mimosa, flytrap, whereas tropic responses are like tendrils turning around something or growing towards sunlight. It's usually like a whole genre, like everything does it. So that's how I sort of keep those differentiated. Okay, back to leaves. We're also running out of time, so I have to pick up my pace here. Um, so I remember I mentioned that we tend to name things that are space in plants. Um, in this instance, we've named the space in between the lobes as the sinus. And you'll read field guides where they talk about the depth of the sinus or how wide the sinus is. And that's just the space between these lobes of this leaf. This is the margin on the edge. These are the lobes. We've got 5 of them on this one. We've got the primary midrib. We've got the apex. We have the base of the leaf, and we still have the petiole. Now we're going to go to compound leaves versus simple leaves. So here's that simple leaf, and we've got that axillary bud next to it. This is a pinnately compound leaf. So we have an axillary bud here, and then we have these things that are coming out opposite each other, and it looks like a feather. And pinnatus is from Latin, it's for feathered. So we have a lot of leaves that have a pinnate compound leaflets. And these are called leaflets. Sorry, I should say that. So this is a whole leaf. And when you get to divided or compound leaves, these are called leaflets. And this whole thing together is called a leaf. And then a palmate leaf has a leaf, has the petiole going up, and then it's got separate leaflets that all come out of one center place, like a palm. And then the hard part is when you're in the field, how do you decide whether it's a simple or compound leaf? I look for swollen petiole bases, and I also look for those axillary buds. So in this one, you can see it's widened right here and fat for the swollen base. So I know that's the start of that leaf right there, which is a compound leaf. And same for this black walnut here. Here is the axillary bud, and I know that all of this out here is going to have all the leaflets, whereas this is the axillary bud, and there's one leaf right there, so that's a simple leaf. And then leaf types. Here's our simple leaf over here. Here's our palmately compound leaf, and here are our pinnately compound leaves. This is once divided, so that's a pinnately compound, and this is our mimosa, so this is twice divided. So that swelling and axillary bud are way down here on the twig. So we have it not once, but twice divided. So we call that bipinnately compound. And I think I have those all there. Oh, and I even put in this one. This is another pinnately compound leaf, but you can see it has wings here, which I think are really cool. I like wings. And that's just sort of demonstrating that at one point there was a simple leaf, and we're either going to simple leaf or we're going to compound. Don't know. From that family, it's the sumac. I think we're going to compound, but that's just me. And then I decided to give you all of the different species here. And when I use an asterisk like this, that usually denotes a non-native or an invasive species. When we're looking at leaves, we also want to look at their attachment to twigs. So the attachment or arrangement is called phyllotaxy. When it's alternate, there is one at each node. And then the node, remember, is the joint at the stem where things normally bear leaves or buds or flowers. Opposite, there's two, one born across from each other at the same node. So that means it's opposite, just like we have one arm on one side and one opposite it. And then whorled is when we have leaves or buds arranged in a whorl around a leaf, around the twig at a node. There's obviously more, 3 or more that are rising from there. And I also, another one that we see in attachment and phyllotaxy for wildflowers is this roseolette, and that, or Basil rosette is another word I also hear for it. Those are also something we hear about when we're talking about arrangement or attachment of leaves. And then we have leaf shapes. There's lots of different leaf shapes. Yeah, it's crazy. There's tons. It's fun to play with these and to talk about them and come up with, whoa, my computer's deciding to go without me. To look at them and come up with different leaves for each one. We won't have time for that today, but there's also, I should caveat this, that botanists love mixing and matching these terms to describe leaves. So they'll call something oblanceolate, and you don't know if that oblong is the top or the bottom, and you have to try and figure it out, which is why a diagram is always helpful. And I have a book on just botanical terminology, and it's phenomenal, and I always recommend it. Then we have leaf tips, venation, and bases that can be different. And these guys here with these long cunate tips, that's usually to help water come off leaves because that's important. And again, you don't have to worry about too much of it. Here's leaf margins. This comes in a diagram that I really couldn't stand, so I finally broke the diagram up and I put everything into order. So here's things that are serrate on this side, and then we have things that are undefined, so sinuate or undulate. And then over here we have ciliate and dentate and crenate. And I think lobate and spiny come back in here too. So this would be a holly, and this would definitely be our oak leaves. So this arrangement works much better for me than the alphabeticalized way it's normally broadcast and shown. So hopefully it helps you, but you'll have to use it some other time. And then we have leaf surfaces. Remember, leaf surfaces are where we have hairs covering a twig or a leaf, and these are dermal tissues. There are all sorts of different trichomes that you can encounter. Some of my favorites are the stellate, where they look like little stars. I don't know why that keeps going crazy. And then here are twigs. Twigs are what we are looking at right now outside. And we have the terminal bud here, and then we have these lateral buds on the side, or axillary buds. We can have leaf scars. And then we have, so this would be a node between this bud and that bud, or the internode, sorry, and these would each be nodes. And then from here to here would be the growth from one year to the next. And there's little dots on here. Those are going to be lenticels. You can find hairs on there. You can even look in these bud scars and see bundle scars, and those would be the vascular bundles that would be in there. Some people talk about the pith. If you have a black walnut, it's a chambered pith. Very interesting stuff. And this is all you really have this time of year. Buds, bark, and twigs. And I think that we may have asked you to bring some twigs with you today. So we'll see if that happened. And we should also differentiate. There can be flower buds versus leaf buds. This is a spice bush, and you can see that the flower buds are much bigger than the leaf buds. That's not always the case, but it is in this one. And this is spice bush. I think they're kind of cool. And there should be more leaf buds than flower buds, but spice bush tends to have a lot of buds and flowers in spring, so it might be equal. And I haven't figured out where this should be best placed in this presentation, but I thought I would quickly do an aside with a dichotomous key. I'm not sure if anybody's ever used one of these. This is what we use to identify a plant, a dichotomous key. Dichotomous means having two equal parts. And this is, as I said, what we use to identify organisms, and it provides a series of choices between A and a B. And those choices are called couplets. And then if you make all the right decisions, it should lead you to the name of your organism that you have. And so that makes a little sense, here is a little tiny dichotomous key that I have thrown in here. So this would be a couplet, leaves broad or leaves needle-like. And but first, before we do this, we need to have somebody pick one of these items on this picture board here for us. Any selections?
How about the one on the right?
The one on the right. This one?
Yes.
All right. Yes. Okay. So leaves broad or leaves needle-like for that plant? Do you want to go to 2 because they're broad, or needle-like and go to 3?
Go to 3.
Go to 3? I would agree with that because they don't look like this or that. 3A, cone woody and elongated or cone soft and round?
You go with soft and round, but it's hard to tell without
Yeah,
touching
it is. And I actually think that I don't like this picture anymore. Whoa. Because soft and round to me, it makes me think of doesn't make me think of cedar. It makes me think of like Atlantic white cedar or a cryptomeria or an arborvitae, not and that's, I guess that's sort of what this is. I think I would have preferred to have a juniper there, but we'll leave that for next time. So that's what a basic dichotomous key looks like. They can be a lot longer and bigger depending on where you start. So if you don't know if it's a gymnosperm or an angiosperm, you have to start way back at the beginning of the key. If you know if it's an angiosperm versus a gymnosperm, like you can get a third of the way through the tree of the key or less, depending if ferns and other things are included. And then something I like to remind people of when you are working with a key, it is something that somebody has made up. So it's artificial. And we want to make sure we remember that. So if you have a habit of picking up lots of different keys, so like you use the flora one day of Virginia, then you decide to use West Virginia's flora, and then North Carolina's flora, all of those authors have a different way of looking at classification. So they would probably drive you crazy because you have to get into the head of the author to really understand how they're using their key. So in this case, you could start this key in several different ways. Do you guys want to give me some options?
Has 4 legs.
Yep, you could start with has 4 legs.
Has slats.
Slats. You could start with alive versus inanimate object. There's lots of ways to look at this. So I like to keep that in mind when I start thinking about doing anything with creating a dichotomous key. So I'm gonna pause for a second because this is where we had another activity for you stuck in here. And I think I was told you guys had picked up twigs for me. Is that right? Oh, good. I'm gonna stop sharing so I can see your twigs. Oh, excellent. All right. Oh, you guys got some good stuff. So we don't have a ton of time, but I would like to take a couple minutes. Actually, first, does anybody need another real break? I'll give you 3 minutes if you need to run to the restroom or something. Okay, I got some shaking heads. Um, Mine's still outside. I'm gonna go get it. That sounds fine. Um, so when we look at these twigs, the first thing we would do if we were trying, so can everybody hold up their twig for me so I can see it? Because we're gonna have to do this together. Okay. Does anybody have a twig that has any live growth on it right now? Do you have needles or scales? No. Okay. So we don't have to worry about angiosperm versus gymnosperm because it sounds like we all picked up things. Oh, Mary, she's
I've got
gone to
one.
go get something.
What's that
Oh,
cherry?
wait,
It's cherry.
Brian
Give some
has one.
cards.
So Brian, it looks like you've got is that rosemary?
It's you.
That's a U. Okay. It's a little hard to tell from here.
Yeah.
So we could start our key if we were writing our own dichotomous key with um evergreen or angiosperm, right? And if it's evergreen, guess what? Brian's comes out first. um, we would actually start it with evergreen with leaves, or without leaves. Oh, and Mary's made it a little harder because she came in with a pine. So we would want to start. We're going to go back a little bit, and we would start with angiosperm or gymnosperm, evergreen with needles or broadleaf. And Brian, I think yours is still broadleaf, even though they're small. Um, and we'd actually probably you would probably still follow the needles thing because it's a U, right? So from our U, we'd have to from our gymnosperm versus angiosperm, needles versus broadleaf, two would have to be um, needles in pastels of 5 to get Mary's because I think hers is a white pine. And then number 2 under that would be smaller needles in singles, and we would get Brian's U, right? Cool. Okay, so we did 1 and we've done 2, but we left that angiosperm, if it's broadleaf, we left that blank, so that would be now number 3. And 3 underneath, we'd have to look at whether we have branches with opposite buds or alternate buds. And if anybody has a world bud, you have to tell me because otherwise we have to do something different. Oh, I think that Phyllis has a red bud twig. Tracy, yours looks like something that has a big fat compound leaf. Is that a hickory of some sort, Tracy? No. Uh-oh. Fig. Aha. You guys really did like to get me on these on these twigs. Um so we would have opposite and branching twig or versus alternate branching twigs. And Amanda's looks like she's got an opposite branching twig. Debbie looks like she has an opposite branching twig. Um, Trisha, yours, I can't tell. Is yours a boxwood? Yeah, okay. Um, so we're gonna hold on to that. I think that's alternate. So we have 2 opposite branching ones. Diana's yours is alternate, I think. Okay, so Debbie and Amanda have opposite branching. Now we'd have to try and distinguish between those opposite branching. Amanda's has red twigs and white buds, is that right?
Yeah.
Yeah.
Yep.
And Debbie's, yours looks like thicker twig, but I'm not really sure from here. Do you have any red on that twig?
No.
No. Okay, so we could use red twig versus brown twig for these opposites to separate them. Um, I think Amanda's might be uh, a maple. Is yours a Japanese maple? I've never looked at the buds on a Japanese maple.
Yeah.
Okay, so I sort of pulled that one out. And then Debbie's, I can't see it. What do your buds look like, Debbie? Are they
The buds themselves, there's like a compound one right here.
a compound bud?
Well, I mean, sorry. So you're talking about where it comes out of the
Well, we already looked at where it comes out. It's opposite branching,
right.
right? So
So
now I want to see what your
the
bud looks like.
So it is, well, I can tell you what it is.
Okay, well, you said you have lots of buds at the end, so that makes me think it's a silver maple, but I could be wrong there.
It's a forsythia.
Oh, wow, I was way off. Okay,
Yeah.
so you're in the okay, but our gray twig versus red twig would have been able to get you guys separated far enough that we could get to give answers on that.
Right.
So I think that's good. So we have a lot of alternate twigs, right? So we have an alternate twig that still has leaves. So Trisha and her boxwood would come out pretty quickly there. Um, Jean, I think Jean or Phyllis had the redbud twig. Is that right? And then um uh oh, Debbie picked up a second twig.
Yeah, I got two too.
Okay. We're not gonna be able to get to your second twigs today. Um but so we have thick twigs and we have thin twigs, right? After we had Trisha's boxwood go out with current leaves on it. Um Diana, how thick is your twig? We have to unmute because I can't hear you.
It's pretty thick. It's not. I also have a Japanese maple, which is a lot thinner than this.
Okay. It looks like you're holding can you tell me what that is?
It's a cherry.
It's a cherry. Okay. And I think Tracy's is going to be the thickest twig that we have here. And she said hers was a fig, right? So we could go with thick, fat twig versus thin, delicate twigs. Would that be enough for you, Diana, to think you'd go with thin, delicate rather than stay with fat and thick twigs?
Okay.
Okay. So we could get Tracy's taken out with the fig right there. And then we would go down and we would have Phyllis's redbud and we would have Diana's, you just told me what this was.
It's a cherry. It's a cherry.
Cherry. Okay, good. Your cherry probably has when you scratch it, do you have a funny smell? If it's a
Yeah,
black cherry.
yeah. Mm-hmm,
Okay.
yeah.
So we could do the next one as a scratch and sniff. So if it smells pungent like burnt almonds, it's cherry. And if it has no smell, it would be Phyllis's redbud.
It has a smell.
Yeah.
Yeah.
So that's how we would write a dichotomous key just for the twigs that you guys brought today. And
mine had a nice smell too because I immediately knew what I picked because it was the spice bush.
Oh, you had a spice bush too.
Yes, I had a spice bush.
Okay, because then we could have done like punctate white lenticel dots all over the twig because it should have those. And you could have been in the scratch and sniff one too. So if we had so if we had done Phyllis, Diana, and Debbie, we would have had scratch and sniff or other. And other would have been Phyllis's redbud. And then we would have had scratch and sniff smells like pledge lemony fresh or smells like for the other one burnt almonds, and then you could have gotten to those two. So but remember, like, this is subjective, right? You know, we had to sort of guess if it's a thick enough twig versus not thick enough. We had to think about our smells. Everybody has different smell opportunities and abilities, so this can be a tricky thing to do. which is why you've got to really, if you decide to get really into this and identifying and using dichotomous keys, I would choose a key and use that continually. Don't do it like I do and pick up 5 different books and use 3 different dichotomous keys, because it's can be very frustrating, because if you don't remember how that person thinks, you're going to go off in the wrong direction, and it's going to be an annoyance rather than a fun sleuthing ride to find the right plant. Okay, let's get back to this presentation. We don't have too far to go. You guys have been doing great. And we're going to do reproduction. Okay, so flowers have different parts to them. The flower is a site of reproduction. It's often used to attract pollinators. A generalized flower has highly modified stem tip and 4 whorls or series of appendages. And when I talk about a whorl, I'm talking about a circle. So the bottom, we have the top of our stem, and the bottom whorl is going to be where we have our sepals. The next whorl above that is going to be our petals. The whorl above that is going to be our stamen, and that's made up of the anther, and then the filament with the anther on top, and the anther is where we're going to have pollen grains. And those pollen grains will have gone through meiosis, not mitosis, so that we have genetic mutations, and we also have 1 copy to add to the ovule when we get there. And then the center piece in the middle is going to be the ovule or the pistil, and it's going to have the ovary with the ovule inside. It's going to have style, and it's going to be the stigma on top, which is sticky to receptive pollen. And I think that women have style, which is why I remember that the pistil is the stigma, style, and ovary. And the stamen is male. It has men in it, so that shouldn't be too hard. That's how I remember it. So I wanted to just show a rendition of how we got from a petal. Like, everything starts as a leaf in a plant, right? And everything has to be modified to get to what we want. So this is a leaf that's become a petal that's slowly turning into an anther on top and a filament on the bottom, so that we get the structure for that piece in the for the male part of the flower. And then likewise, here is the pistil. We would have had a modified leaf that had sporangia on the edge, and that would have rolled up and fused, and then started getting smaller and smaller. And that's how we get the ovary. And then we would have the style and the stigma from that's coming out of the top of it. So that's how we got the female part of the flower. And then here's the whole reproductive process, where we have whether it's diploid or haploid. And I'm not going to go through all this because we don't have time today, but I wanted to make sure it was in all of your materials for this class. You're going to get a copy of this presentation at the end. So I should have said that a lot sooner than now. Sorry. We also want to look at the ovary position. We want to see if it's inferior or if it's superior. So superior is going to be above all of those parts that we just talked about. Inferior is going to be below all those parts. And then we have some where it's in between. We call that perigynous. And we see that a lot in the cherries. And then another thing we have to talk about when we talk about flowers is symmetry. If it has radial symmetry, that's actinomorphic or regular. And if it has bilateral symmetry, that's irregular, and it's zygomorphic. And it's considered that if you have irregular symmetry, then you're a more advanced flower than if you have radial symmetry. That works pretty well, especially if you think of the monocots. You get to the orchid flower that's very specialized and very zygomorphic, but radial symmetry still applies to our asters, so sort of mix on that one. Then we have flower terminology. Our angiosperms, they can be perfect flowers where they have both male and female parts and they're capable of reproduction within. They can be a complete flower. That means it has all four whorls. It has the sepals, petals, stamen, and pistils all in one flower. And then we can have imperfect or unisexual flowers where it only has male parts or only female. And I've made a table of that so that you can see that. So here's the perfect flower. We call it hermaphroditic because it has both male and female parts. And then we have staminate flowers for male flowers and pistillate flowers for female plants. And then we have some percentages of how that breaks down in case you're interested. And then I wanted to show you some staminate flowers. This is our red maple, and it has these beautiful, I think they almost look like fireworks, staminate flowers where they have all of these filaments with the anthers on top and pollen ready to go. A lot of people think these are wind pollinated, but I do see lots of insects on them when they come out. And then over here we have the female pistillate flowers, and you have these two pieces here. This is going to be the receptive stigma at the top of the style, and the ovary is going to be down in here somewhere. And these are really helpful when you see these two pieces. That tells you how many carpels or ovaries this plant has. And you'll remember that when you see the samara for a maple, it usually has that two-winged helicopter. So that means this is two carpellate. And the next thing I want to show you is that we have monoecious versus dioecious. Monoecious is one house, and that has male and female flowers on the same plant. And then we have dioecious, where we have male and female flowers. They're both on separate plants. And a great example of that is holly. We have male plants that do not produce berries, and we have female plants that do produce berries. And red maple is sneaky. It can be dioecious or it can be polygamous, where it actually has male and female flowers on the same tree. And then it can also have separate male and female trees. So there's always exceptions to all the rules. And that's the same here in botany. And then this black birch that I have here, these are the male flowers hanging down, and then these are the female flowers hanging that are heading upwards. But since both of them are on the same tree, that's a monoecious tree. And it's really interesting. I learned recently that this does not describe, monoecious and dioecious do not describe something like a magnolia, where both male and female parts are in the same flower on that tree, which kind of blew my mind, but I was corrected, and then I looked it up, and sure enough, there's separate flowers on the same tree for monoecious. So separate, imperfect flowers on the same tree, and they can be complete or incomplete. Okay, and then inflorescence. So this is a shoot bearing a cluster of flowers. So if you look at my diagram, you will see that that does not include a trillium because that is a solitary flower. But it does include the black haw, which has hundreds of flowers on this stalk here. So there are two different parts of an inflorescence. There's the peduncle, which is this piece here, and then each flower is on a pedicel. And you'll hear that funny term peduncle when we talk about fruit and when we talk about, yeah, it's usually a stalk bearing a fruit as well. It comes up twice in botany, and not sure why they use the same one, but that's okay. And then we have different inflorescence types. And this can get really confusing, so I didn't go into too many of these. But I wanted to make it clear that there are so many. Um, this is the inflorescence spike where all the flowers are attached to the main stem without stalks. So this little cartoon diagram shows how it does. And the size of these circles, and whether there are circles or not, depends on how shows the order in which the flowering happens. So the smallest circle flowers first, then the larger circles, and so on. So those are spikes. Racemes, their flowers are not directly attached to that main stem. They have their own pedicel or stalk from the main stem. So you can see that. And then panicles, these are racemes. They're attached to the main stalk. So each of these, this one, this one, this one, this one, this one, this one, those are all different racemes that are part of this. And I'm not sure what that is over there. And then we have umbels, queen ends lace. So here's our main stalk. And then we have flower stalks arising from one point on the tip of the stem. And then they're usually in a flat head on top. And the bloom is from inside out for those. Catkins, we just looked at the black birch. Here's the river birch, and we have these catkins. These are flowering arrangements that are cylindrical spikes, and they're usually drooping and pendulous. They lack petals. They're usually single sex, and they're usually wind pollinated. The other one I have in here is the white oak. These catkins are much longer, less flowers. And not as dense and tight as the river birch, but they are considered catkins as well, which is why I wanted to put them in here. And again, because we have catkins here and we have female flowers in these leaf axils here, this would be considered monoecious. And then we have the composite head. So this is where we have two different types of flowers. We have, they're all smushed together on this top here. On the inside, we have disc flowers, and on the edge, we have these things called ray flowers. So there are two different types of flowers smushed together. And this is the sunflower family or the Asteraceae family, used to be a composite family. And you can see in here, here are the ray flowers, and in the center you can see little tiny flowers that are the disc flowers. So when you eat a sunflower seed, it's usually from the disc flowers and not the ray flowers. And you can also have some composites that are just ray flowers or just disc flowers. And can you guys tell me an example of either one of those? We're almost done, but you're not off the hook yet. Do you guys think you need any help? Okay, so a dandelion is all ray flowers. And a thistle is all disc flowers. And when I did work in Florida, there was even a rayless sunflower. So that would be considered all disc flowers. Why they still call it a sunflower, totally not fair, but there you go. I had bones to pick with some of those people, but I'm trying to be nice. Okay, so the next thing that we should talk about is spathe and spadix flowers, because several of you today had examples of these when we were looking at our houseplants. I wanted to show you one that is out there right now. The skunk cabbage is out, and this is a spathe and spadix flower. So the spathe is actually the um, bract that surrounds the spadix. The spadix is where all the flowers are. And uh, I like skunk cabbage a lot, so here's another picture. And the spadix is in the center, and it's covered with lots of florets of fleshy tepals. And we haven't talked about tepals, but tepals are an anagram of sepals and petals. So that's why we have sepals. I didn't do that, someone else did that. The florets initially have the female stigma receptive to pollen before the stamens emerge and open to release pollen. So they're separating by time when the stigma is receptive to pollen to prevent self-pollination. They're fascinating, by the way. And we should talk about adaptations that flowers have for pollination. So there can be color, odor, nectar, flower orientation, landing places, nectar guides, whether the flower opens at day or night. And then we talked about that symmetry earlier. And these are all the pollinators that you can have: bees, wasps, butterflies, flies, moths, beetles, hummingbirds, ants, bats, small reptiles, and mammals. And recently they discovered an amphibian that is pollinating flowers. So quite impressive list. And one of those is really cool. We're going back to the skunk cabbage. This plant creates its own heat. And it's actively respiring from its spadix. And the putrid smell of rotting meat, which the heat carries out, will attract insects such as honeybees, flesh flies, carrion flies, waterleaf beetles, predators, and predator spiders. The plant also provides them warmth, refuge from the cold, and while inside, the insects can pollinate the flower, which is really, really cool. But in a whole other of phenology, another whole study of botany biology, and this is Aldo Leopold. The skunk cabbage blooms about 1 week before the Eastern phoebe arrives. And the phoebe will go to the skunk cabbage because it's the only thing open to get its food, and it will help it it'll save it from starving during cold snaps when things become scarce for the phoebe. And Aldo Leopold noticed this when he was documenting the phenology of when things arrived and when they bloomed. And you can see that the bloom time of the skunk cabbage has moved up about a month or so. And this is from Wisconsin, these dates, so don't assume that they apply here. But that Phoebe is still just a week behind in returning for that. And we don't have time to get into that today, so sorry, that's all the excitement you get on that piece. The other thing is that pollen does awesome things. And in this plant, it is spring-loaded with those anthers and filaments pulled back as the flower opens. And when an insect lands on it or something disturbs it, the pollen is catapulted out of the mountain laurel. And I'm not sure if you saw that, but I'll play it one more time. There it goes. It's flinging away, which I think is super cool. Catapult launches the pollen at more than 400 times the acceleration due to gravity, reaching top speeds of about 8 miles per hour. And the last thing we have to talk about is fruit. I'm really not going to spend a lot of time on this because I'm tired and so are probably all of you, but we have some slides about fruit in here for you to look at your leisure. And then I thought we should talk about adaptations for dispersal of that fruit. We have gravity, wind, water, fire, explosion. animals moving things around, and sticky hairs and stiff hooks. I just read in the Potomac Appalachian Trail Club newsletter that dispersal on animal fur is not a common strategy for seed movement. Less than 5% of all seed-bearing plants produce burrs. And I gotta say, I think it's a whole lot more than that, but I didn't do the research, I just saw the note. But I thought that was really interesting. Um, and then the next thing I wanted to show you is the eliasome. These are plants that produce seeds that have a fleshy structure on it that attracts ants. The ants take the seed back to their home underground, and they eat the eliasome and they throw the seed into their dump where it germinates and starts to grow and new plants happen. Fascinating. And these are three plants that have eliasomes. It's kind of a new discovery. There's probably lots more, but that's all I had time to find for you today. And here's the tools I think you need if you're going to go out and do any uh, looking for plants yourself. I think you should have a hand lens, a ruler, a tree ID book or any ID book, some pruners, and take all those things with you. You'll have a great time. Here are my references, and there is questions if you have any for me. And I think that is it. We made it to the end. You guys did a great job. I'm really sorry. I really took all that time.
Thank you, Emily. You were wonderful. I think that was just for a Zoom, for changing your whole presentation to Zoom today. That was amazing. I mean, because it's hard, but that's great. So if there aren't any questions for Emily, I just want to have a closing announcement before we sign off, before anyone leaves us today. So certainly appreciate, Emily, you're sending us these slides as well.
Thank you.
Anybody need Emily for questions? My only announcement is about next week, of course. Since we did such a great job being able to deal with the snow